Okay. Officially, good morning. MED110, Anatomy and Physiology 1. This is Dr. Garais. This is Wednesday morning, June 10, 2020. And what is due today? Your task, discussion, and of course, lesson. And if you haven't done so yet, I'll give you till closing of business today. And my closing of business is around 6 p.m. But you know me, I'm always looking at my phone and checking stuff. So what are we doing this week? Let's look at task 3. This is, we're doing cytology, which is the study of cells and cellular biology and organelles. And let's go back to this picture that we had before. If you remember this picture, it went like something like this. Maybe I should have just not clicked on that. Here you go. If you look at this picture, right, we already looked at some atoms, molecules, and we looked at macromolecules. Remember the carbohydrates, right, which are composed of carbon, hydrogen, oxygen, and proteins, carbon, hydrogen, oxygen, and nitrogen, fats, carbon, hydrogen, oxygen. So you can see that our organism, us, the human being, is comprised of some very basic things. Well, the atoms get together, make molecules, molecules make macromolecules, and these macromolecules, they eventually make cells. But the cells, you can look at them as kind of like a separate entity. Let me lower my fan. Okay. All right, so let's now look at the cell kind of like a mini human being. And inside the cell, there are these things called organelles, which are like organs, but they're for the cell. So it's like a little microcosm or a little mini world that we have. And it makes sense because if this mini world, the cells, goes down, who's going to go down next? Tissues, organs, then organ systems, then my patient is sick. Okay, so we're actually kind of semi-following this this train of thought, which is kind of related also. Do you see how it's like arrows and boxes? It's kind of like a flow chart, and it's also mimics your concept maps. So this is kind of the scientific way to look at things. So now we know because why we're looking at something. Let's look at, and that's part of cell theory. So you could watch these, and if they're what do you call that? If they're not, if the links aren't work, I haven't checked these since last term. Please give me a call so I can give you alternative links and whatnot. But pretty much alternative links. Well, just tell me it. So because I have to inform IT anyway. Now, if we look at these Carolina notes cells, and we're still in task 3, they have. Don't worry about the. They have all these descriptions of what pretty much the structure, which is anatomy, and the function of all of these things. And we're going to be going through all of these things momentarily, which is a nice summary. So let us now go through this, and let's now look at your typical cell. And let's get one that has, we'll try to get one that has like all the, yeah, this one I remember has all the parts in it. Pinterest, hate you. This one. Oh, this is cute. This is a nice little flashcards where you can test yourself. But that's not what I want. I want just the picture. Is it so difficult just to get a picture? Is this the one I want? I like this one. Wait, kill this, open this, find this, find that. So if we look at this, oh man, there might be even greater explanations. Oh, wonderful. Oh, cool. Maybe I'll put this this booklet on. If you look at the cell, it's surrounded by a cell membrane, right? Which is like a skin. And then you have all these things on the inside, which are like its organs. Okay. Now, the cell membrane we're going to be talking about last because it's a little bit more complicated. Well, it is like a skin, but we're going to be talking a little bit more detail. But the cell membrane is also known as the gatekeeper, because the gatekeeper, it's going to allow certain things in and it's going to let certain things out. Now, the brains of the operation is this, the nucleus. Okay. Well, let's go through this book. It's the first time I've ever seen this book, and I find it neat. So let's look at the cell, of course. Now, you may wonder how all of these things stay in place, because inside the cell, it's kind of like a jelly-like material called your cytoplasm, okay? And of course, the cell membrane keeps everything in, and the cell membrane, like we just stated, is our gatekeeper. So it keeps all the stuff that we want on the outside and all the things we have on the inside. Now, there is also the brains of the operation is the nucleus, and that also has a skin or a protective membrane or a protective covering. So you could see when we talk more about the nucleus in a moment, that this is a pretty important feature of the cell, because that's where the cell is going to hold all of its blueprints on what is exactly its job and how it's organized. And that blueprint is in a tightly coiled bunch of proteins called your DNA, deoxyribonucleic acid, right? It is the blueprint. And what I mean by blueprint, I mean everything. Like if you're gonna like Cheerios, if you're gonna have heart disease, are you gonna be tall? Are you gonna be short? It actually, for all intents and purposes, how are you to be born and how are you to die, right? All of it in this nice little circle, which, by the way, this cell, right, is, of course, microscopic. Now, within the nucleus, I stated that there's, of course, envelopes, and they've got little holes to let some things in, to let some things out. Now, within the nucleolus, now the nucleus is like the whole thing, but the nucleolus, you could see how all of this stuff is called chromatin. It's all the DNA, but all the DNA that we don't need right now, that gets even more tightly packed in here. And this should blow your mind, because this has every single piece of information of who you are, right? So when they do, like, you know, on criminals and stuff, or not on criminals, or even a paternity test where they swab your cheek cells, those cells that we get, and then we look at the nucleus, and we look at the DNA. This is the thing that we're looking at. We're looking at the DNA. And when we're looking at genetic problems, remember we talked about atoms, macromolecules, and things of that nature. Genetic diseases, they start here. Because if the blueprint is smudged or mistaken or just simply wrong, what's going to happen? All the other things down the line that we talked about, organelles, cell, tissues, organs, systems, all of that's going to be messed up. So this is the nucleus is the brains of the operation. It directs everything, and it has DNA inside of it, and the less tightly packed DNA is called chromatin, and the really tightly packed stuff is called the nucleolus. And how does it really tightly pack itself? Remember we talked about hydrogen bonds? They're not that strong, and they're loose, and they can be bent and shaped. Well, that's how we get the nucleolus here. Now, nuclear envelope, nuclear pores, and again, I have to reiterate, These pores, they let some things in, let some things out. And later in this lecture, and also continuing to next week, we have to let some of that information out. We have to let some of that information from the DNA out, and it gets released through something called your RNA. So it goes from DNA here, then it gets less condensed chromatin, right? And then from that DNA, it gets copied and then get released into the world through these nuclear pores. And then if you see here, this is endoplasmic reticulum, and they have these little bumps. Each one of these bumps is called a ribosome. And the DNA gets transcribed onto RNA. The RNA then leaves this nucleus and then goes here to this endoplasmic reticulum, also known as the rough endoplasmic reticulum, also known as the RER. And it's called rough because it's got these little bumps on it. And all these little bumps are ribosomes. And that RNA gets then translated to the ribosomes and makes proteins. And we're going to talk about that all in greater detail. So to recap, because that's a lot, you have the nucleolus, highly concentrated DNA. The chromatin, it's still concentrated DNA, but it's relaxed a little bit. Then the DNA here gets copied onto RNA, right? DNA is deoxyribonucleic acid. That's the blueprint. And I want you to look at the RNA as kind of like a messenger or a foreman that will now transcribe and then translate the genetic code onto this rough endoplasmic reticulum. And on this rough endoplasmic reticulum, which is a protein factory, it has all these ribosomes, which you could see them as like protein like factory workers. And they're the ones who are gonna get out the proper protein in the proper sequence, in the proper code, because of the message that came from the DNA to the RNA and then to this rough endoplasmic reticulum. And you could see how complex that is, but yet it's a beautiful package and it makes sense. And if you look at it like a story, then you'll remember the nucleolus, chromatin, nuclear envelope, nuclear pore, endoplasmic reticulum, rough endoplasmic reticulum to be specific, and these little bumps called ribosomes. And you could see, isn't that the same exact way that we're looking at regarding, concept maps? It's like a story. One thing leads to another, leads to another, leads to another. And isn't that how your memory works? You kind of remember things through a chain of events. And this is one set of chain of events that should amaze you because it's happening every millisecond of your life, even before you were born. And all of it requires energy, like we talked about. And the second that energy stops, the second you stop eating, the second you stop existing, all of this also stops. Now the ribosomes, see the little bumps on this rough endoplasmic reticulum? They are protein, they're part of the protein factory. So I want you to think about this rough endoplasmic reticulum as a protein factory, and the ribosomes are the protein builders or the protein workers within that factory. Now, ribosomes also can be found just floating around, but the bulk of them are in this rough endoplasmic reticulum. And you can see here, this is an endoplasmic reticulum. Now, the rough endoplasmic reticulum makes proteins. What does the smooth endoplasmic reticulum do? Well, if you look at the rough endoplasmic reticulum, it has a whole bunch of ribosomes. So you know that this makes proteins. Now, remember we talked about fats, carbohydrates, and all the other things? Well, that gets processed and made here in the smooth endoplasmic reticulum. And they call it the smooth or the SER, excuse me. They call it the SER, the smooth, because it doesn't have any ribosomes, okay? But the same exact thing happens. You have the DNA to the RNA, sends the message to the smooth endoplasmic reticulum, hey, go make me some carbohydrate, go make me some fat, okay? Now, what happens when it makes me some proteins and makes me some fat? Well, we got to transport this stuff the within uh, the cell itself, right? Let's go back to this picture. So let's say I make something. So it's gonna be pinched off in this thing called a vesicle. So it's gonna be like even like a mini cell, but it doesn't really, it's just a package and it floats around and it gets pinched off by either this envelope here, which is your cell membrane, the outer. So if it gets pinched off from the cell membrane, it gets released out into the world. But we also use these vesicles to communicate proteins and carbohydrates and sugars and whatnot in between all the other organelles, okay? And remember the cytoplasm that we talked about? The cytoplasm that we talked about also has like this microtubule scaffolding. So it forms like these little like highways or stairways where these vesicles can then float towards their intended target. And that should also equally amaze you because remember we talked about specificity regarding receptors. There chemically there's it has to match up with everything like a lock and key, and that's how you know that the hormone is going in the right place or the protein is going to the right place. There is a lock and key so that it doesn't go to the wrong door, and we do it through these vesicles. Another thing that these vesicles also carry, let's see if they'll talk about it. Right? Well, let's go back. There's this thing called a lysosome. Where is it? Here. Now, a lysosome is just like this. It's a vesicle. But if you look at the word lysosome, lys means to break down, some means body. So inside these specific vesicles are materials that will break down things. So if we're looking at this, sometimes there's garbage and waste products floating around. We can't have that. So we pinch it off in vesicles, and then the lysosomes come by and they what? Eat out or process the garbage, and then they could eject it outside the cell. So if you look at it, and remember, the pinching off can go in and things can go out. So if you look at it, it's like a human being. It's got a brain. It processes things inside these things, right? And then it does what? Eats. Glucose goes in, and what goes out? Byproducts, right? Oxygen can go in, and what can come out? Carbon dioxide. And that is, of course, called respiration. So vesicles form a number of things, and lysosomes, and another type of lysosome is called a peroxisome. It's like peroxide, because peroxide also does what? Breaks down stuff. If you've ever had like a wound and you pour some peroxide in it, you see it like starts sizzling and it starts like eating away at all the necrotic tissue. And that's the purpose of why you put peroxide on a wound. Now, how do these vesicles, these things right here, get made and processed? Well, they get made and processed through something called the Golgi apparatus. Now, the Golgi apparatus, right, is lysosomes digest things. I was ahead of the game here, but the Golgi apparatus is, let's see if they have it here. Here's a Golgi body or Golgi apparatus. They kind of look like smooth endoplasmic reticulum, but if you'll see here, the Golgi body or the Golgi apparatus starts pinching off vesicles here. And the Golgi apparatus is the kind of like the post office of your cell. It's the thing that packages it and sends it on its way and sends it to wherever the target of that vesicle should be. So if it's a lysosome, the target would be to eat up byproducts of the cell and take out the garbage, right? If it's maybe if it's a hormone, it'll be taken to the end of the cell or the cell wall to be released out into the external surface of the cell so it can do its job outside of the cell. So, and all of this, see if I can, There you go. All of this requires energy. And remember, we talked about the two things that your body loves for energy is oxygen and glucose. Well, guess what? This stuff called the mitochondria use to make energy. It uses oxygen and glucose. Oopsies. I wanted to get to this. So here are these like, let's look at a better picture. I want a better picture. Here you go. There's these bean-like structures in your body and with all these little folds, and it takes in oxygen and broken down glucose, and then it creates something called adenosine triphosphate. Now, ATP looks like this. Right? Looks really complicated, but isn't, right? Now, what's ATP? All it is an adenosine group, which is this, right? And it's connected through 3 phosphate groups. If you can see here, here's one phosphate, right? Here's another one. Here's another one, PO3, right? Well, these bonds right here, right? This bond, this bond, this bond, these bonds right here are high energy bonds. So when you break down or hydrolyze ATP and you break it down to ADP, and a hydrolysis reaction is just introducing water into the reaction, right? And we now know what water does, which its special properties. It can separate things like sodium and chloride. Well, guess what it can do? A hydrolysis reaction requiring water can now separate the bonds. And then once it separates the bonds, it releases energy, okay? So ATP, or adenosine triphosphate, gets broken down into ADP, adenosine diphosphate, by all the organelles in your cell to make energy, so that, you know, it can do stuff. So it can make the proteins, store the DNA, translate and transcribe the RNA, so it can do all of that. So that's why the mitochondria is known as the powerhouse of your cell, because it requires fuel, oxygen and glucose, and it's going to create power. And the power comes in the form of this molecular fuel called ATP, or adenosine triphosphate. Look, I'm ahead of myself. Okay. Been teaching this too long. Now, let's not, there's another part in your thing that go over plants, but we're interested in humans right now. So, but looking at all that, and then you could compare it to all of this, now you could see that this is every little cell is its own little person with organs that can go wrong, right? And also it has these other things, centrioles, cilia, flagella. We'll be talking about these momentarily when we talk about the plasma membrane or the cell wall. Okay? So there's a lot, right? But if you look at it, it's just like a little mini human being, and it's nice that you're, well, you have this video and these notes go through all of that, as you can see here. Now, phospholipid bilayer, right? And what's a phospholipid? Lipid means fat. Phospho is phosphorus. That's all it means. But the thing about the phospholipid bilayer, let's look at that. Let's look at a phospholipid bilayer. And I love this picture. I've been using this picture forever. Let's see. There you go. Right? You see this? Let's make this bigger. Oh, good. You see, this is your cell wall. Okay? It's not just the skin. Well, the skin is also equally complicated, but you can see here it forms 2 layers, and it forms this phospholipid bilayer so that it kind of keeps the water out here, right? These are phosphorus heads. These are hydrophobic, meaning that it doesn't like water. It will push water away. So it makes sense that you keep the water here on the outside, and the water that's on the inside, And remember I told you about the cytoplasm having these little microtubules, like these little highways and little stairways? This is how we know this is the inside of your cell, because there's a cytoskeletal filament. They're made up of little mini microtubules. They're little mini microproteins, and they give the cell the shape, and they also form the little highways that if I have vesicles and things, they can travel around here. And if you recall, if you look at that picture of that vesicle, right? Oh, I already closed it. Remember, it had a skin that looked just like this, right? Right. This is what I love about anatomy and physiology. A lot of connections, a lot of Illuminati stuff going on all the time. So all of this, all these little heads, these phosphorus heads that are in reddish orange here, those are hydrophobic. Now, these here, these lipid chains, right? They form together, and you see how they're all connected here, and it forms a double wall of phospholipid bilayer. I'm sorry, I think I said something backwards. Scratch that. The phosphorus heads are hydrophilic. They love water, and that's why there's water out here and there's water in there. So scratch what I just said. I did it backwards. So let's scratch that. Phosphorus heads, I want you to think hydrophilic, right? And they love water because there's water out here and there's water in here. Now, we all know that fat or lipids, and these lipid chains are here, right? And you can even see there's cholesterol here as well. Those are fat soluble. That means they're hydrophobic. So now it forms a layer that water can't easily go in and out. Now, how does things go in and out of a cell? Well, see these blue things here? These are membrane proteins, and they form protein channels. Another thing that these proteins do is they also provide strength to this phospholipid bilayer. So you also have integral proteins here as well. So you could see how this is definitely called the gatekeeper, because it's going to let some things in and it's going to let some things out. Now, how does it do that? How does it know what to let in and know what to let out? If you look at this glycoprotein up here, that means it's a protein connected to some sort of sugar. And a glycolipid here, it's some sort of fat connected to a sugar. That's all that it means if you use your medical terminology powers. Now, the glycoprotein coat gives the cell its identity chemically, so that the body can recognize it either as itself, or the body can recognize it as one of its employees, right? Now, in an autoimmune problem, your body doesn't recognize these glycoproteins, so it treats this cell as foreign, and then it'll start eating it up, which is bad news. Now, this glycolipid as well, along with these glycoproteins, not only for identification, glycoproteins and glycolipids also serve as a lock and key mechanism for these protein channels. And one classic example is insulin. Now, sugar is a 6-carbon sugar. It's big. It won't go through here, right? Which, by the way, for all of you wearing masks, virus would be the size of, I don't know, a little bit smaller than that dot on the word lipid. So do you think that can go right through the bilipid layer? Yep. And the one other frightening thing about viruses, they're ubiquitous. Many of them can use RNA and DNA, and it uses your RNA and uses your DNA. So mask up all day, but there are other things that our body does to protect ourselves, and masks are, you know, just an extra precaution. But honestly, if someone posted a very funny meme on my wall the other day. It's like it's like trying to catch a mosquito with a chain link fence. It won't work. There are other ways to protect your patient. But yes, masks, and also socially and psychologically, the mask, at least it's something for our patient to do to at least feel semi-protected. But you can see now things like the size of a bacteria, which may be the size of this pointer right here, can't really go through. And especially if this door is closed because of this glycolipid and glycoprotein lock, there'll be a lid here, and then bacteria, you know, won't be able to go as easy. Same thing also with other foreign body, because this is the gatekeeper. It's going to keep things out. Now, my example that I did before I digress was insulin. And insulin is a hormone, and I want you to think of it as a key that will unlock a door. Why? Remember we talked about the mitochondria needs glucose? Well, the glucose is big. It can't come in. The mitochondria is in the inside. It's not on the outside. So what does insulin do? It attaches on to this glycolipid, right? And then that will tell these protein channels, hey, open up a door so that the glucose can come in. So there'll be a bigger protein channel, and this big glucose molecule can now go in, now can go to the mitochondria, and now the mitochondria can make energy. Okay, so that is what's happening every time you have a meal. Now, what happens in diabetes? Now, where does the fuel belong? It belongs inside here in the mitochondria. And I made the analogy before, I think it was this class, where diabetes mellitus is, that's when the fuel is on the outside. And the analogy I made was, it's like pouring gasoline, like, you know, when you're at the pump, instead of putting it inside your gas tank, you start pouring gasoline on your seats, on the hood of your car, on the roof of the car. That gasoline now is very, very dangerous. But when it's in the fuel tank, in the mitochondria, that's where it belongs. And that's what diabetes mellitus really is. It is, these sugar molecules are all floating all out here and doing a heck of a lot of damage, because they're big, they're bulky, and they're not doing its job, which is to provide fuel for the mitochondria. And if you look at it like a lock and key, right? Insulin-dependent diabetes mellitus, it's a problem with the key or the insulin. Non-insulin-dependent diabetes mellitus or diabetes mellitus type 2, it's the problem with this, the lock. And think about it. We as Americans, especially myself, I live on pretty much Wendy's consistently, even before the quarantine. So what happens if I keep on abusing this lock? The lock will get broken, right? And that's also one of the other theories of type 2 diabetes, because now we're seeing it in children, a ton of children. When I was in training 20 years ago, I think in a room full of 40, I'd see one kid who was obese, who was clinically obese, and maybe out of 100, maybe one or two kids that had diabetes mellitus type 2 or even type 1. But nowadays, you go into any pediatric ward, according to the National Kidney Foundation, which I'm a staunch member on. I'm always giving them money and going to their seminars. It's scary. It's almost 42% of kids, and the incidence rate is climbing and climbing and climbing for juvenile diabetes, especially juvenile type 2, which is scary, because that means a lot of it is what do you call that? Is diet dependent. Okay, and I sometimes question how much of diabetes mellitus type 2 is really genetic, because when you think about it, who taught you how to eat, right? it goes, and also behavior is also part of genetics as well. So if you remember the old days, well, at least my old days, growing up in the 70s, my parents always used to say, hey, there are starving children in the Philippines, clean your plate. Well, I don't raise my kids that way anymore, because I tell them the second you feel full, just put whatever you got in the refrigerator, right? The second you even remotely feel full, don't eat. If my kid wants to miss breakfast every once in a while, I don't force it. I go, sure, pick up a snack later. And Suffice it to say, it's good to report right now, I have children ranging from 21 to 6 months. Only the baby, the 6 month is a little bit on the heavy side, but all the rest of them uh, aren't going down the same path that I'm going down, because because of this system right here, okay, of letting things in and out. Now, one more thing, clinical thing I want to talk about, letting things in or out. Now, most diabetes mellitus type 2 patients, right, don't need insulin. They only need insulin when they're really bad. You know what you need? Not only diet control, right, portion control, so that you don't have that much glucose, but there are other protein channels, and they're called GLUT2 and GLUT3 protein channels in your cell. They open up automatically to let glucose in when you exercise, at least 20 to 30 minutes, 3 times a day, according to who's those people? Um, American Heart Association, the AHA, right? And they're thinking of increasing that number to 40 minutes, 3 times a week. But typically, it's 3 times a week, 30 minutes, and I mean 30 minutes, not just sweating. You know, if you've ever been to a gym and see people who look like they're doing more socializing than working out. I mean, really, 30 minutes of barely being able to talk and really, really pushing yourself. So you could see just right off the bat how important the gatekeeper is. Now, let's mention this cholesterol here, because I have to mention what we all know as the good cholesterol or the bad cholesterol. You could see here there's cholesterol embedded naturally within, but there are also protein channels, and cholesterol is actually a it's a way to get fat in and out of your body. And you have high density lipoproteins, and those are ways to get cholesterol out of the system. And that's why we love HDLs. It's not that it's bad or good cholesterol, it's just that the HDL loves to transport things out of the body. So it's a nice measurement of how well are you metabolizing, your fats. Now, your LDL and your VLDL, which is your low density lipoprotein or your very low density lipoprotein, those are the transport systems that gets fat into the system, right? So you could see now in a laboratory setting how, because knowing your cell and knowing how it works can it can lead you to clinical understanding. Okay, so that is the lipid bilayer. Okay, integral proteins is for the stabilization of the wall. You have your channel proteins for communication, to let things in, let things out. You have cholesterol, not only for structure, but also as a vehicle to move fat in or fat out. You have your glycoprotein and your glycolipids for not only identification chemically of what the cell is, but also as a lock to open up these channels. And of course, the phospholipid bilayer. And again, I must correct myself. The phosphorus heads up here are hydrophilic. So there's water on the outside, water on the inside, and these long lipid chains here in between, they are hydrophobic or fat soluble, which makes sense because you can see now the cholesterol is now embedded within on the inside of this phospholipid bilayer. All right. And that is your cell membrane. We took it a little bit further than you know, what you may have done in high school or um, previous lectures. And again, all the stuff that I was talking about, it's all in here. Now, let's talk about osmosis and diffusion. Well, you saw the lipid bilayer. That is a semi-permeable or selective permeable membrane. And if something passes through it, that's called osmosis. Now, what's diffusion? Diffusion is any particle in this world wants to be free, wants to spread out. And a classic example of diffusion is, let's say you're at Macy's and you're at the perfume counter, right? If I spray, you know, whatever lovely perfume I have at the counter, right? What happens? If I spray it right there, all the molecules will want to do what? They'll want to spread out. And they'll keep on spreading out until when? Until I can't even smell the original perfume anymore. And that's also how things that are in water molecules, things that are in water work. And you can also do that with, you know, a drop of food coloring. If you just put a drop of food coloring in a very large, you know, pitcher, right? Just a drop. In the first couple of seconds, you see a high concentration, but then little by little, it expands until you don't even notice the color anymore because it's so dilute. And that's essentially how your body moves fluids. It moves fluids from a region of high concentration to a region of low concentration. Now, there are some times where we want to go the exact opposite, from a region of low concentration to a region of high concentration, but that requires energy. Osmosis and diffusion do not require energy. Therefore, it always goes from a region of high concentration to a region of low concentration. And it'll form different kinds of solutions. So if we have solutions that are even, right? These solutions look kind of even, right? The number of big dots and the number of little dots kind of even out. That's equilibrium. That's the same. Iso means the same. Isotonic. Okay? Now, human beings, we're made out of not only water, like 66% water, we're also made out of salts. So our normal solution, or our isotonic solution for a human being, is 0.9% sodium chloride solution, also known as NSS, or normal saline solution. And that's when all the fluids are all under equilibrium. Now, if you pretend this is like a bilipid selectable membrane, if you look at these little white circles as water molecules, and you look at these larger black circles as, like, I don't know, like Kool-Aid, the water molecules is called the solvent, and the Kool-Aid, or the powder parts, is called the solute, right? Hence the term solution. Now you can see this solution on this side, right, has way more Kool-Aid than this one. But what will eventually happen in this semi-permeable membrane? It will go to equilibrium eventually, right? It'll all even out. But you could see, why does it even out naturally? You could see that these Kool-Aid or solute particles are moving from an area of high concentration to an area of low concentration. So then these things will then move where? From left to right, and then to make equilibrium. Now, why is this important to us and us clinically? Well, this forms an osmotic pressure. And it's the ability, and we don't do this lab anymore because honestly, it doesn't work. But we could see how osmotic pressure works. So this is how it works. Now, instead of Kool-Aid, let's look at these solute, or these little black molecules. Let's say that's salt, right? or sodium chloride, okay, within solution. Now, do you see what will happen if I have a high concentration of salt here, a low concentration of salt here? There'll be osmosis through this bilipid layer, and then it's going to start moving this way. That's going to form a greater osmotic pressure, and it's going to also shove water in the direction that the salt is going. Now, this is really important for hypertension. Now, that's why we're always telling our patient, hey, cool it with the salt, even though it makes things taste awesome. Because the salt, if you have more salt in your system, as you can see here, you can see how more crowded everything is. And what will that do to the pressure? It will increase. And then you drink a couple of beers along with your nachos, and that's water. What will happen to your blood pressure? It will increase, and it will create a hypertonic solution. So you could see wherever salt goes, water goes because of the osmotic pressure. And that's also the same reason why in the bar, if you ever, I don't drink anymore, which is a shame. I miss it dearly. But for health reasons, you know, when you get older, you really shouldn't, or really should lower it down. Well, I used to love bar food, but I only love it with beer. Because if you ever tasted bar food by itself, it's very salty, very sugary. It doesn't taste that good. But if you got it with a beer, oh, or you have it with alcohol, what will it do? Oh, it makes you want to drink. You know, those free pretzels and the little snacks that the bartender puts out for you. The bartender isn't stupid. Wants you to drink more, wants more tips. So they put out these salty things, and wherever salt goes, fluid will follow. So the salt makes you dehydrated, then you drink alcohol, it makes you more dehydrated, then you drink more alcohol, and that's a problem. And that's a big problem, especially now it's beach weather. I've been in the entertainment industry for 30 years and working in bars and stuff like that. I used to do Atlantic City yearly in the summertime in July and August. And how many people pass out on the beach because they have those ridiculously salty, ridiculously sugary foods. Then they combine it with alcohol. And then what happens? It goes, you have increased osmotic pressure, increased blood pressure. Then they pass out. And it's a nice little cocktail for disaster on a typical Sunday afternoon. So with that being said, that's osmosis and that's diffusion. And the difference between the two is osmosis deals with, what do you call that? Diffusion through a semi-permeable, semi-selective membrane. And we got all of that from this cell structure and function in your task. So when we look at your tasks, like what are you supposed to be submitting a concept map, you could talk about the cell structure, organelles, and the functions is what we did today. And what's this, these tutorials? Oh, you could look at this. This is nice to know. It's comparing prokaryote versus eukaryote versus a virus. This is real quick. Try that at home, right? Ugh, ignore. Love telemarketers. You could also have these little tutorials, but this is the main in task 3. This is the main stuff where it can it provides a lovely summary of all these things going on, and or you could use, of course, your textbook as well. But a quick synopsis of prokaryote, eukaryote, and viruses. Like I mentioned, viruses are very, very small. They are actually parasites. They don't have any DNA or RNA of their own, and they insert themselves inside the nucleus, and then they hijack your stuff. A classic example is HIV. HIV is a reverse transcriptase, meaning it will take your RNA, reverse it, and back it up to the DNA to tell the DNA, hey, we're not making your proteins anymore. We're just making viruses. And it's really hard because the virus is very small. It gets inside the cell, so it's like a perfect home invasion. So from the outside, your white blood cells and your immune system think everything's cool, right? But on the inside, it's a virus-making machine. And by the time that cell implodes and then the viruses get free and then invade other cells, it's too late. And that's why there's a prodromal phase where someone could test negative for HIV. And oh, by the way, that's another scary thing that people don't want to talk about is that's the same thing with the testing. I've been talking about it for like the last 3 months. I've been asking colleagues, how do you know there's no prodromal problems with your testing? How accurate is your testing? Right now, I have a colleague of mine. She heads up pediatrics in a hospital in New York City. She has 59 COVID cases, and she's questioning. She has 59 pediatric cases. All of them got sent home. All of them are either symptomatic or asymptomatic, but no one went to the ICU. No one went to NICU, right? So it begs the question, right? What is the diagnostic criteria? Do these kids really have COVID? Okay, it's one of the, again, makes me sound like a conspiracy theorist, which I'm borderline am at this point. Now, prokaryotic versus eukaryotic, right? Oh, by the way, regarding reinfection, can a virus reinfect you? Yes, because remember, it's ubiquitous. It can use RNA, it can use DNA, and it can hijack whatever cell it wants. Now, as long as the person is not immunocompromised. If the person doesn't have any other problems like kidney problems or any other illnesses, you'll relatively stay healthy, right? But a lot of these kids that I just mentioned, and that's a lot in the last month or so, 59, that census is a little high for pediatrics, but it is what it is. Prokaryote versus eukaryote. Now, who are you? You are eukaryotic cell. Everything that we just talked about in this lecture is eukaryotic cell. So look at the prokaryotic cell, which is, bacteria and other things of that nature. But if bacteria is similar in fashion to eukaryotic cells, you can see why antibiotics not only affect, the thing that they're trying to kill, they can also affect you in adverse ways. And as an internist, I really don't like meds, because medications, you're toying around with the most complicated chemistry set, which is the human being. So just briefly look over there, and there's a similar synopsis of what I gave a moment ago. So that's one thing that's due by next week, of course, your task, right? And many of you are doing it smart. Like, oh, I'm going to do one picture or one concept map, just cell structure, one concept map, just organelles, one concept map of functions of organelles. But I'm trying to challenge you. Try to put all 3 of those pictures together and see if you can link them, and you could see how they are linked. So it makes you like one big picture. Okay. Now, what does the discussion for this week look like? And I could give you a hint. And if you still have issues with APA, please give me a call so I can give you my mini video that I made regarding on how I see and how I do APA format. So you watch this this video on mitochondrial disease, and you discuss how it goes. How is it affected? Like, how does the signs and symptoms relate to the mitochondria? Hint, hint, we already know what the mitochondria does, right? So somewhere in your introduction, you have to talk about what does the mitochondria do? If I have damage to it, as in this particular disease, what will lack of ATP do and present itself as signs and symptoms? And again, APA citation, please get it from a legitimate source, .gov, .edu, .org. I hate cutting points on you guys who are doing some good research just because you got it from a not so academic source. And please look at the comments. Some of you just ignore the comments. Please don't ignore the comments, because then your grade will stay the same. But if you attack the comments and go, oh, okay, let me give Dr. Garais what he wants, then it gives me it gives me a reason to give you full credit. Okay. And that's my goal here. I want to give everyone full credit, but I need the criteria of what is required. And remember, at least 200 words. Make sure it has an introduction or your opinion in an introduction form, a rationale on why you think what you believe is the answer, and then the most important part: supply evidence. Get me the source that will back up the things that you say. All right. And I will be a broken record regarding this every week. Every week we're talking about it until we get it right. Because your other professors, especially in your humanities courses, they won't tell you anything. They just chop points. And if you keep on doing it, they see it as plagiarism and they will report you, and then you're going to have some trouble. And I don't, and I don't want that. I want you guys to know why your professor does stuff. Now, regarding lesson 3, there's a lovely video here and more of the same stuff that I talked about. Again, lysosome, more lovely stuff, right? But what do you need to do? Don't do the lab. Again, don't do the lab, right? We already went over osmosis and diffusion, but these are, again, reiteration of the notes, nice notes. But what do you need to do for the lesson? Remember, you click on this, download the case study, click download the case study, and then you have part 1, then you have part 1 questions, part 2, then part 2 questions, part 3, and then fill out this thing, which, by the way, can you just copy and paste and then cite that you got it from the notes? Yeah, why not? Right? So do parts 1, 2, and 3. That should be good enough, right? And it kind of relates to the discussion, how defective cellular organelles can actually then downstream present this disease. So that's it for the week. If there are any questions, comments, recipes, if not, I now have to, what am I supposed to be doing now? I think I have to go take care of my children before my mother gets buried. All right. Any questions? Are you good?
Yeah, I'm all set. Thank you.
All right. Do what you need to do, and I'll see you next week. Have a good one.
All right, you too. Bye-bye.